Welcome, everyone. Thank you for joining the GlyGen demo. So today we will be doing, or I'll be doing, a GlyGen short demo, and this is the first of the series. And my name is Raja Mazumdar. So myself and Dr. Michael T. Meyer, we are the PIs for the GlyGen project. GlyGen is a common NIH Common Fund supported project. And what I will be showing today are the websites and the new tools that GlyGen has recently developed. But before I get into that, there are a few things I want you all to pay attention to. So what we are trying to do is a live Q&A. So what it means is that while I'm giving the demo, it's a really short demo. So it's 20 minutes and then 5 minutes of Q&A after that. But during my demo, you can ask questions. So what you need to do on your WebEx, most of you are very familiar with WebEx, most probably, or something similar like Zoom. So there is a chat box that you can activate by clicking on chat, bottom right-hand corner. And your interface might look a little bit different. So, but you will see something like this. And what you need to do is that you need to select all panelists and then write your question. And then in the background, Renee or Jeet or Amanda, who are co-hosting this demo with me, will be able to respond to your questions or answer your questions in a Google Doc, which we will bring up end of the demo and go over. Another way to contact us would be through the Contact Us form. So if you want to send us an email through this form, then you have to go to the GlyGen website. Under Help, there is Contact Us, and you can send in your comments or your questions. Either way, we will be monitoring both of these mechanisms of getting in touch with us today, and we will get back to you. After the demo, we will be sending out a survey, and we really hope that you will take a minute or so. I think it takes a minute to finish that survey to let us know what you think, if there are other types of or other things on GlyGen that you want us to demo. This is first of a series of 3 demos. That's our plan. So next month, most probably, we will have another demo. These are really short demos. So we can only show very specific things in GlyGen. And this demo is being recorded. You all registered through the Eventbrite. So we will be sending you a recording of the demo. And with that email, you will also get a link to the survey. So Glycans, the glycan diversity is immense. And this is a paper published in 2012 by Kelly Mormon, Mike T. Meyer, and what this figure tells us, the huge diversity. And it's well known that glycoconjugates, whether secreted from cells or found at the surface or inside of cells, regulate essential functions in health and disease. Almost all currently marketed biologic pharmaceuticals are glycoproteins. Glycan structural features influence glycoconjugate functions. And the big question is that, although it is so important, where is this data captured? Can it be harvested in a way that is informative and that may reveal new knowledge? And if you look at other omic sciences, so you have nucleic acids, genomics, you have proteins, proteomics, lipidomics, and so on. But what about glycans? What can we do in terms, or what we can say in terms of resources that can help us better understand the role of glycans in health and disease? So if you look at it, all but one of the major classes of biomolecules that form the 4 building blocks of life are described by robust databases. So what GlyGen is doing is trying to fill this gap. Now, the types of data is vast. So in GlyGen, we are in our 4th year of the project, and we have decided to focus on organism by organism. So what we have done in GlyGen, so as of today, in GlyGen, you have human, mouse, rat, HCV1A, and of course SARS-CoV-1 and CoV-2, and synthetic glycans. We do plan to add additional organisms over time, but we wanted to have a robust data model, and I'll show you why it is important to have a robust data model before we start putting all of the data together and integrate it. We have to do it in a certain way so that people can query it to get knowledge. So today's demo, as I said, will be very specific on a certain aspect of GlyGen. There are many other things you can do in GlyGen, but I want to present to you something that is quite interesting to me. So N-link glycosylation is actually a really nice model to look at role of mutations or single nucleotide polymorphisms, SNPs, and connect it to function. So how can we connect genomics data with proteins and glycans? So this model allows us, and we'll give you a flavor of what one can do. So here's an amino acid sequence, EGFR, and on this asparagine, you have this sugar, this glycan attached to it. So the N-link glycans attached to asparagine's in the sequence context of ASN, asparagine X can be any amino acid except proline, and then serine or threonine. Now, if you mutate this asparagine to something else, some of the amino acid, this glycan will not be attached to it. So you'll have a loss of a glycosylation sequence or a loss of a glycosylation site. Now, in 2005, there was a really interesting paper that was published, which talks about gains of glycosylation comprise an unexpectedly large group of pathogenic mutations. And what this paper showed, and this is directly from the paper, Mendelian susceptibility to mycobacterial disease, who are homozygous with respect to a missense mutation in IFNGR2, creates a new glycosylation site. So an asparagine is added and it forms a new site. And in that paper, they used HGMD, which is a commercial database, to scan the human genome. And HGMD has a lot of GWAS type of studies to allow that. So they predicted gain of glycosylation, predicted loss of glycosylation. This was in 2005. And currently, we have extended through another project called Oncomics, which is an NCI ITCR project, which supports similar type of analysis, gain or loss of glycosylation and other PTMs. And GlyGen absorbs this data, and I'll show you what it does. But again, so in 2005, this paper was published. A decade goes by. In 2019, in Nature, this paper was published, which pretty much shows the effect of that new glycosylation site. So what it shows that people who have that polymorphism have this new site where a glycan is attached, which introduces steric clashes and which results in the protein complex not forming correctly. So the basis of the disease was explored further through this paper from an initial paper which was published in 2005. And actually I found this paper when I was preparing this talk on GlyGen, because I knew the 2005 paper, I did not know about this paper. So that's the power I want to show, how you can explore knowledge through data integration. And all of this information is coming from different places, by the way. And we tag all the evidence. Here is a screenshot of that particular site in GlyGen, where we show there's a T to N polymorphism. And then this is the disease that is associated with it. So there are I wanted to have the acknowledgement slide somewhere in the middle just to make sure that I have this slide up. There are several people involved in GlyGen at in different ways and at different levels. I'm going to now jump directly into the demo. So the easiest way to get into GlyGen is trying to see what a protein entry has, what kind of information a protein entry has. And I have been using this as an example because all of us use some kind of a browser, web browser. I'm using Google here. You can use something else. But if you just type in your gene name, space, and GlyGen, usually the first hit is the protein of your interest. So with that, I'm going to go directly to Google and do this search. The rest of the slides, I have them. I'm not going to show because I'm going to do it directly as a demo. So I'm going to type in EGFR, GlyGen, and I get this hit. And by the way, we are recording this demo, and we will send you the link so that you can take a look at it, because some of the tasks that I will be doing will be a little bit fast. If you have trouble following, you can always review the demo. So this is a protein entry. On the left-hand side, there are menu items which you can click on, or you can scroll down this page. This is a long page. And that's why we have these menu items. Of course, the most interesting one are the glycans that are attached to this protein. So here is an example where we show that different glycans are attached in different positions. So this is position 56. There are, why is 56 repeated many times? Is because there are different evidences which tells us that this glycan is coming from the resource GlyConnect, which is a collaboration with the group in Switzerland. And PubMed, they have evidence, the papers that are associated with it. So if I scroll down to any of these entries, I can, of course, click on the publications that are associated with it. Unicarb KB is another collaborator of ours. And you can click on the site. And what we provide is a site-specific view, which I think over time is going to get really powerful, and I'll show how and why. So in this example, we have N, remember I said NXST, so K is in the middle, and then T, and this asparagine has this sugar, and there are two papers which talk about this finding. At the same time, we also integrated data from UniProt KB, which does not provide the structures, but they have really valuable information about publications associated with that position and is talking about N-glycosylation. So you have multiple papers which have evidence pointing toward that particular position and what is known about that site. So this is an example how we can explore the different sites. So now I'm going to go and show the next item in my demo, which is the site search. So you see these new tags. So these are the new items that we have recently added in GlyGen. So in this site search, we are going to try to find, let's say, you can look at any of these residues, you know, alanine, cysteine, and so on. And here I'm going to select asparagine because I'm still following the same theme, the N-link glycosylation. And now you can select, or well, I want to find all asparagines which have glycosylation information. I also want to find how many of those sites have mutagenesis information and search. And if I click on the search protein site, it will pull up all the sites which have N-linked glycosylation. And also there are some mutational studies associated with that site. So this is the table that I got. We have some kind of a score. You have a start position, end position, and glycosylation and mutagenesis. It happens so there is also single nucleotide variation from cancer research, somatic mutations, which we also capture. And in this example, for example, we can click on one entry, and this entry is Naga gene. And in that particular protein, there are different glycans that are associated with it. And what we can do is that we can look at the mutagenesis information, and we see here this N to Q. There's a loss of glycosylation site that has been observed. You can also look at single nucleotide variations also. So this is the site search that I wanted to show you. The next I want to show is a super search. The super search is very novel, and I want to spend a little bit of time talking about this figure. And this figure actually is a live figure, and it has numbers on it. This gives you a snapshot of entire GlyGen, a view. So there are these many proteins, 64,000 proteins. Out of those proteins, there are 33,000 glycans associated with them. There are sites, protein sites, 430,000 protein sites. There are 12,000 enzymes which synthesizes the or associated with synthesizing the glycan, or several proteins, 91 which are synthesizing the glycans, and there are several proteins which are enzymes. Many of them are associated with diseases. Many of these proteins, of course, not many, almost all of them are connected to gene information, and then these proteins come from certain organisms. And earlier on in my second slide, I showed there are 7 such organisms that we are working on right now. Many of these glycans have motifs that are associated with them, or we have been able to assign to them, and you can then explore the motifs. Clicking on any of these entries will allow you to do a site or protein specific search. So here is an example. If I were to click on the protein, I can say I want to search for uniprot KB accession. I can say, or ID, EGFR, human, and when I do a search, I will get all the proteins, or only one protein with all the glycans associated with it. So the same way, so one protein, there are 105 glycans, 105 sites with 32 glycans associated with this particular protein, which is EGFR. Now, if I were to do a site search, I can also do a site search similar to the one that I showed you, but let's try to do this or look at the results in a slightly different way. So I can say amino acid, N, glycosylation, true, I will, and then mutagenesis, true, and single nucleotide variation, also true. So when I do this type of search, I will end up with a list of proteins which satisfies this query. And from there, I can take a look. So here, there are 107 such proteins with 176 sites, but let's look at the list of glycans. So these proteins, which glycans are associated with these sites, which have been shown by people, by researchers, that when you mutate them, you see some functional impact, or maybe in some cases do not see that impact, but nonetheless, there is data associated with mutation or data associated with variation in cancer. So in this example, because I'm now looking at the results in a glycan-centric view, I can now click on the glycan ID, and I will get a list of or get the entry which I clicked on, and it gives me the accession numbers, it gives me the mass, the glycan type, the organisms that this has been found in or associated with, the motif, associated proteins, and so on, and the biosynthetic enzymes, subsumption, and more. So that was a brief demo of the super search. The last thing that I want to show you today is the GlyGen mapper. It's an ID mapping tool, and often you may have a list of glycans, glycan IDs that you want to map, and if you have let's say glytucan IDs. So and then you want to know what PubChem compound IDs I can map them to. So you have glytucan, these are glycans, you have glytucan IDs, and you're trying to map them to PubChem. You can just put them here in this text box, or you can load a file. So once you load the file, I have approximately 500 or so glytucan, 510. And here you see that all these were mapped to PubChem compound ID. So these are PubChem compound IDs. And we have glytucan IDs which can map to PubChem, or KEBI, or other types of IDs that you are working with. So these are some of the examples that I am showing today. We are getting close to the Q&A session. I will pause here and see if there are any questions that you might have. So if you haven't typed in your questions in the chat box, please go ahead and type your question in the chat box, any comments, and I will try to answer them in the next 5 minutes. After that, if some of the questions I cannot answer right away, we will send a reply to everybody who registered for this demo, the responses to any of the questions that have come through because of today's demo. So let me go and see if any questions have been posted. So let's see, Q&A. All right, so there are some questions that we already added. Some of these were already added by us based on questions that we have received previously, not necessarily for today's demo. So I'm going to go through them. So question number 1 is,
Roger?
Yes.
Sorry to interrupt, but we have some questions coming up in the chat.
Right.
So we will try to add them in the question, and I think you can also read it.
Okay. All right, I can open up the chat box in parallel and see. It might be a little bit tricky to do this live on the chat. I will try to see what I have here. Why don't I start with the mouse ortholog and then go from there. So the question was, how can I find orthologs of protein? So if I go to glygen.org, or actually we can start with EGFR GlyGen. And while I'm doing this, by the way, Jeet, Rahi, and Renee are responding to some of the questions directly in the Google Doc or chat box. It's actually in the chat box itself. So Jeet, Rahi, and Renee are responding to some of your questions while I'm doing a live demo within a demo. So this is how one can look for orthologs. So this is the EGFR protein. We have the term homologs here, and for those of you who really delve into orthology, orthologs, paralogs, and homologs, it can get tricky sometimes. What are the true orthologs of one protein? But we get many of this data from the group who are involved in defining orthologs. And in this particular case, example, there are two proteins. One is from, I clicked on homologs from EGFR, and based on the calculations from OMA, we have this following entry from rat, which is an ortholog, and this following entry from mouse, which is also mapped as, or we, for argument's sake, let's call them homologs, but most likely they are orthologs, to really, if you do not find your anything here, for example, so for certain proteins or genes, you may not find any homologs. For that, most likely it has not been possible for the groups that we get the data from to identify homologs for that protein in the organisms of interest. So when you click on the organism, in this case, this is a rat, you can see the different pieces of information. Another important thing that you can see here, when you look at the sequence, you can look at the different sequence, for example, for N-linked glycosylation, even though in some of the model organisms, there is not enough data to show glycosylation data. By looking at the sequence, you can quickly see how they look like. You can then align this to the human record and try to see if it is possible, if any of the annotation from the human can be transferred to your rat protein. So let me go back to the example here. Do you plan to integrate some tools that can also build 3D structure of glycans? And the answer is yes. So we are working with Dr. Rob Woods of UGA to integrate, and we hope to have the integration done by the end of this year. Don't hold me to it. I think that's our plan. But yes, Dr. Woods is part of the GlyGen team, and we do plan to have that. And yeah, you're familiar with the glycan sequences. So yes, the answer is yes. Are you planning to expand the data for fungi and bacteria? The answer is yes, but I do not know if we will be able to do it by next year. So our next organism in the pipeline is Drosophila. And with the SARS, we added SARS, and we are exploring, because every time we add an organism, it's not trivial. We have to get the entire proteome. First, we have to define the proteome with collaborators at UniProt and EBI and others. And then we start adding. And also, when you have multiple organisms for the same bacteria, multiple strains and isolates, how to represent that data is something that we are still trying to work our way through. So again, the idea is sustainability, the ability to expand and not redo everything. And also, we do believe that the glycans connected to genes and proteins and genome has a really high value. So we want to make sure that we work with other groups, including gene ontology, Panther, and other groups also, to make sure that we are doing this correctly. All those glycan information on GlyGen are based on published results, right? The answer is yes. What organisms do you plan to include? Okay, so I also already answered that. It is 10:59, so I'm going to stop now. Really appreciate everybody joining the call, and please fill in the survey, because based on the survey, we can send you information about our next GlyGen demo, short demo, 20-minute demo, 5-minute stop. The next one will be most probably by Mike T. Meyer, who is the other PI for GlyGen. And he will be talking about some really interesting developments and things you can do with GlyGen. Thank you again. Bye, everybody.